Hello and good morning. Just a few announcements before we start today's topic. There will be a quiz today. I will open the quiz for a whole day so everybody has a chance to take the quiz. However, there will be only one attempt, so whenever you take it, that's your only attempt to take the quiz. It will be time bound, so you will have, as usual, you will have 15 minutes to complete your quiz whenever you sign in and do the quiz. Okay. Another thing is I have been getting a lot of emails about the schedule for the remaining semester. I am following the already submitted schedule, the one that you have. So we will follow the exact dates for our tests, and final is of course going to be the same as well. So this will be your last quiz. Then you will have another test. I think, yes, test 3. That's towards the end of April. You have 2 of your assignments that are due this month. Your first assignment is your written assignment that is due first, and then your audio assignment, your recorded assignment, is due later. I did it so that you have some time to make your slides and record your audio, your presentation. And that's going to be a 5-minute presentation. Other than that, as far as the topics are concerned, so I am following the schedule that I sent you. It's tentative because I don't know usually how far I would be able to go in a given day. So we will follow those guidelines that you already have. Since you will not be presenting, I had set aside 4 classes for the presentations, group presentations, since there won't be any group presentations. So whatever we are far behind, we will catch up during those extra lectures. So use your tentative schedule as a guideline for the rest of the semester. Okay, and with that, I will start today's topic. So today we're going to talk about translation. We talked about DNA replication, talked about transcription. So next step is that DNA that has been transcribed, those genes that have been transcribed, those need to be translated in order for that gene to be functional. So that's what the functional genomics we talk about. So translation is the synthesis of protein, right? So we will be today we will be focusing on a genetic code, second genetic code, translation initiation in both prokaryotes and eukaryotes, and a part of elongation. Okay, so these materials are from Watson 7th edition, your book, chapter number 15 and 16. And there I have given here page numbers for both 15 and 16 that you might want to read in more detail. For optional material, you might want to read this paper from 2006, A Unified View of the Initiation of Protein Synthesis. It's a really good review paper to understand more about translation. Okay, so with that said, let's talk about translation. Translation is production of a polypeptide chain from an RNA template. This RNA template is has been transcribed from a DNA template. That's your gene, right? Translation always precedes 5 to 3 prime on the RNA, so will always read 5 to 3 prime of the RNA. Protein synthesis occurs from or you can say that the 5 prime of the mRNA will make the amino terminus of the protein, and the 3 prime will make the carboxyl terminus of the protein. So when we talk about protein, we usually talk about N terminal, the amino terminus, and C terminal, the carboxyl terminus. C terminus is towards the end of the protein. N terminus is towards the beginning of the protein. The genetic code that you all know, it refers to the 3 base codon that designates specific amino acid. So a codon will tell you which amino acid need to be added here. So your mRNA sequence will tell you what the protein sequence will be. So you can deduce that protein sequence from your mRNA sequence based on the codons. An open reading frame, that is, or ORF, is the protein coding region. This sometimes is also called cistron. Cistron is, if you want So cistron is really an alternative term for gene. It's a section of DNA or RNA molecule that codes for specific polypeptide. Okay, so it's an ORF, is similar to gene, similar to cistron. Usually the first amino acid will be the first amino acid is methionine. The start codon will be AUG for that methionine. Sometimes it's GUG in some organisms. There are 3 stop codons: UGA, UAG, and UAA. Whenever these codons are in the sequence, that's where the protein synthesis will stop, because there is no tRNA or there is no amino acid that codes for these codons. Prokaryotic mRNAs are usually polycistronic. Eukaryotic mRNAs are usually monocistronic, but they can be polycistronic in some cases. There are a few examples of polycistronic mRNA in eukaryotes. Prokaryotes mRNA are mostly polycistronic, which means that the same gene, just there several mRNAs are together for multiple genes. They have same promoter region. Okay, so here this is an example. You can see this is a eukaryotic example because there's 5' cap. Remember, this 5' cap is not present. The 5' caps or poly A tails are not present in prokaryotes. So you can see, if you look at the structure of mRNA, you would know that this is a eukaryotic mRNA. This eukaryotic mRNA has this the first codon that will bring the first amino acid is usually AUG. So this AUG will code for methionine. This methionine will be added here, and then it will make the rest of the sequence, whatever that sequence is, whatever those codons are, those amino acid will be added, resulting in an amino acid chain here, a peptide chain here. This is the stop, so it will stop here. Okay. This is an example of a prokaryotic translation. So here, this is this translation. This is a specific sequence that will be recognized. This is the start site. The start site will make this polypeptide, this peptide here. This because it's polycistronic, so it will keep making this next peptide here, next peptide here. Each have their own start site, but they do not completely dissociate the machinery. Once it starts here, it will just keep going from this start site to that start site to this start site. All right. So this is an example of a monocistronic mRNA. This is an example of polycistronic mRNA. Okay. Other things to consider are codons do not overlap. There are no gaps in codons. So if this is an mRNA molecule, this is first. If this is the first codon, the next 3 will be the second codon, and so on and so forth. These will not overlap, and if there is a change, we talked about indels and insertion and deletion. So if there is a change, then that can change the codon, and that will change the protein as well. So this is a good example here. This inserts and stop. That means this protein will not be synthesized if there is a stop in the beginning. The code is degenerate, which means that most amino acids are encoded by more than one codon. So more than one codon can code for the same amino acid. So here you can see the example. This is leucine. Leucine is coded by these multiple codons. So 6 of these codons will code for the same amino acid. So multiple amino acids can be multiple codons can be used to code for one amino acid. Methionine is the start codon. It's encoded by one codon, and rest every amino acid can be coded by more than one codon. These degenerate codons, they differ mostly in the third position, and that is called as a wobble position. So here, if you look at this, most of the time your first amino acid, the first base pair will be fixed, will be the same. The second is usually the same as well. The third varies quite a bit. So here in the example of leucine, again you can see that this U is conserved. This C is mostly conserved, is either a C or a U, but this the third position is variable. That's called wobble position. Amino acids are specified by these degenerate codons. They do not require a different tRNA for each codon. That means that the tRNA if there are 20 amino acids, there are 20 tRNA as well, one for each amino acid. These are not based on the 64 codons, so they are not 64 tRNA, rather they are 20 tRNA for 20 amino acids. Okay. I introduced this term wobble hypothesis. What wobble hypothesis suggests is that 5' position of anticodon, that will be your 3' position of the codon, right, is constrained as is not as constrained as the other two nucleotides. So this allow unusual base pairing with the 3' nucleotide of the codon, and that's why we do not need as many tRNA as the codons. Ribosomes translate RNA concurrently with transcription in prokaryotes. So this is an example in prokaryotes. So this is ribosome. It's making this mRNA, and at the side by side, this ribosome is making translating this mRNA. And this is possible because remember, prokaryotes don't have nucleus, so they have their whole machinery is right there. So they do not have to send the mRNA to the cytoplasm in order to go in the endoplasmic reticulum and then make those the translation begins there on the ribosomes on the endoplasmic reticulum. In prokaryotes, everything is all together in the cell, so it takes place at the same time. So here, this is an example of this ribosome moving along the mRNA transcript and making this peptide. Okay. This is not the case in eukaryotes. In eukaryotes, transcription happens in the nucleus and translation happens in the cytoplasm. Okay. In eukaryotes, translation occurs in the cytoplasm. It occurs in the endoplasmic reticulum. Usually 2 to 4 amino acids per second are added onto the chain in eukaryotes, whereas translation is much faster in prokaryotes, about 20 amino acids per second. Ribosomes are, this is your ribosome, that's where the protein synthesis occurs. Ribosomes are complex of rRNA, ribosomal RNA and proteins. Ribosomal RNA makes about 60% of the mass of the ribosome. Ribosomes have 3 tRNA binding sites, so both eukaryotes and prokaryotes have these 3 mRNA binding, 3 tRNA binding sites. A site, B site, and E site. A is called amino acyl tRNA site, B is peptidyl tRNA site, and E is exit. So the tRNA will come and bind here, and there is if this the growing chain, there will be another one already here. This will make the peptide bond with the one existing in the P. This will then be pushed into exit and will exit the ribosome, and the one then in A will be pushed into peptidyl, because this will be the next one to make the peptide bond. This tRNA functions as an adapter molecule. It converts the nucleic acid information into protein information, because tRNAs have anticodons that are complementary to the codon on mRNA. So mRNA's sequence, mRNA's codon will be read by the anticodon of tRNA, and depending on what the codon is on mRNA, the tRNA that codes for particular amino acid corresponding to that codon will come and bind. Okay, So you all are familiar with tRNA structure. You probably have seen it multiple times, this cloverleaf structure that most of the time when we talk about RNA secondary structures, you will see this structure, right? In this tertiary structure, it further folds onto itself to make this inverted L-shaped structure. So this is the cloverleaf structure. This is the L-shaped structure. This is, you can see that this is the same loops are here, right here. There are several parts of tRNA, anticodon loop and anticodon. This is that anticodon, the three nucleotides that are complementary to different codons. This anticodon will find the codon and bind to that anticodon. Okay. This anticodon loop here. This is acceptor arm. That's also very important. This ACC is very important because the amino acid will bind to this A position here, and we will talk about this in detail. So tRNA processing in prokaryotes, the 5 to 3 prime ends of immature tRNA. So this is the 5 prime. So if you think about it as a linear molecule, then this will be like this. This will be a linear molecule. But this, because there's enough homology enough complementarity between the sequences, between the nucleotides, so this folds over. And remember, in RNA, wherever there is a complementary sequence, it will bind to that complementary sequence, make these bonds, make double-stranded structure, and wherever it can't, it will make a loop. So here, so these are the nucleotides that are complementary. They will make these structures. So in prokaryotes, this 5 to 3 prime. So this is 5 prime. This is 3 prime. Ends of immature tRNA are trimmed by specific exonucleases, so that they have exact sequence that is required. This is part of the tRNA processing. Remember, we talked about mRNA processing, especially in eukaryotes, where they have to have these different steps: the 5 prime cap, 3 prime poly-A tail, and remove all the introns. So that's the processing of mRNA in eukaryotes. We didn't talk much about processing of other RNAs. So here, this is what we are talking about now. It's in prokaryotes, these 5 prime and 3 prime ends of immature tRNAs are trimmed by specific exonucleases. In eukaryotes, there is usually an intron in anticodon loop. This need to be spliced out before this is a mature tRNA. This involves endonuclease and ligase activity. These endonuclease and ligase activities are provided by an associated processing complex for tRNA processing complex. In both prokaryotes and eukaryotes, this specific enzyme, tRNA nucleotidal transferase, this adds non-template CCA right here, the CCA to the 3 prime ends. So this CCA is common to all tRNAs. So remember, this tRNA structure varies according to what anticodon is, so that it can fit exactly when in the ribosome. So the ribosome is looking for the right fit related to the codon-anticodon symmetry. So if anticodon-codon are aligned well, then it will look for this the structural alignment. So the tRNA sequences vary from according to what they are going to code for. Okay, but the CCA is common to all, and they all make this cloverleaf structure, as well as the tertiary inverted L-shaped structure. Okay, rRNA, or ribosomal RNA processing. So ribosomal RNA is also produced as an immature primary transcript, and it undergoes endonuclease cleavage to form mature forms. Okay. So we talked about the genetic code. The genetic code is, or the codon. This codon has to be correct in order for a correct protein to be made. But then there is also, you can consider a secondary or second genetic code, which is really is tRNA charging. So what does that mean is, depending on what anticodon is, correct amino acid should be attached to this tRNA. So that means this tRNA should be charged with the correct amino acid, because on the mRNA we will the mRNA tRNA interaction will be looking at the codon and anticodon match. But if there is a wrong amino acid attached to this tRNA, that can change the sequence as well, or that can bring incorrect amino acid in the in the growing chain. So this is the second genetic code. So tRNA charging or formation of amino acyl tRNA. Here, amino acyl tRNA will be formed here, amino acid tRNA. It involves the addition of correct amino acid to the 3' hydroxyl of correct tRNA by amino acyl tRNA synthase. That's the enzyme that will add that amino acid onto this, the 3' end. There's a one amino acid tRNA synthase enzyme for each amino acid, which will determine which amino acid need to be added on this tRNA. This activity must be accurate, or else the first code won't work, and the ribosomes can only read the anticodon loop. So it is really important to have a correct amino acid attached to the tRNA, because it will not discriminate against the mischarged tRNA. If anticodon is correct, then it will let the tRNA add that amino acid. Okay. So this the reaction, how this amino acid is added to the tRNA, it requires ATP hydrolysis. So here, this is your amino acid. This is N-terminal. This is C-terminal of the amino acid. This amino acid is first, it requires ATP hydrolysis to activate the amino acid, which so you can see here, this is an ATP molecule. This ATP molecule will bind to this amino acid. It will activate the amino acid. That will release a pyrophosphate. So this pyrophosphate is released. This AMP molecule is now attached to this amino acid, making it adenylated amino acid, because this AMP is there. This adenylated amino acid will then because now it's called a charged amino acid. So here, so it's ATP plus this amino acid will make amino acid plus AMP plus amino acid, or adenylated amino acid, and pyrophosphate. This adenylated amino acid will then bind to tRNA, making it amino acid and tRNA, and AMP will be released from this reaction. So here, this is the tRNA. This is 3' hydroxyl end of this this A here on this 3' end of the tRNA. This will then lead to it will make this high energy bond here. This is now this tRNA is charged, and this is AMP is released from here in this reaction. The shape of tRNA acceptor stem. So this is the acceptor stem is right here, and the anticodon loop right there. These are the most important to ensure that correct tRNA binds to its corresponding enzyme. Okay. All right. So now we are going to talk about how translation initiates in both prokaryotes and eukaryotes. We will start with prokaryotes. So we know that all three RNA classes will be involved in translation. mRNA is the one that brings the message, brings the information of what proteins need to be made. rRNA makes the ribosomes. That's rRNA. In the ribosomes will bind to this mRNA. That's where in the ribosome is where protein synthesis will happen. And tRNA is the transfer RNA that will bring the amino acid according to the codon on the mRNA, so that the protein can be synthesized. And we already talked about that, this tRNA, how the tRNA loads this amino acid onto itself. It's onto the acceptor arm, and this is an energy consuming process. So this tRNA has the amino acid associated to it, it's attached to it, depending on what anticodon sequence is there. Initiation in prokaryotes, it has there's several components that are required for initiation in prokaryotes. So this is the ribosomes, the large subunit and small subunit. mRNA will bind to this ribosome here, and then tRNA will come and bind to these positions. Ribosomes in prokaryotes is 70S. This is prokaryotic ribosome, 70S. It has a large subunit, a 50S subunit, and a small 30S subunit. 50S subunit further consists of two subunits, 23S and 5S. 30S subunit consists of 16S RNA. Now, eukaryotic ribosome is 80S ribosome, 60S and 40S, two subunits here. So, in addition to ribosome, the large and small subunit, we need initiation factors. There are three main initiation factors in prokaryotes and several in eukaryotes. In prokaryotes, initiation factor 1 protects the A site. Remember this A site here. Initiation factor 2 assists formation of initiation complex at the start site. It has GTPase activity. IF2 binds to IF1 on A site. IF3 binds to 30S subunit, so the smaller subunit, and it promotes dissociation of 50S and it should be 50S. Sorry, 50S ribosome into small and large subunit. No, this should be 70S. Sorry, change it. So a dissociation of 70S ribosomes into small and large subunits. So please correct it. It should be 60. It shouldn't be 60. It should be 70. Because the whole initiation complex starts on the small subunit, the large subunit comes and binds later, so it needs to be dissociated and stay away from large subunit until it's required to be bound. And this function is carried out by IF3, initiation factor 3. Ribosomal particles, these particles are denoted according to their sedimentation coefficient, and this S means Swedberg unit. So S in the ribosome stands for Swedberg, which is named after Dr. Theodore Swedberg, who won a Nobel Prize for his discovery of these ribosomes and ribosomal sedimentation. The formula for this is S equals V over A, where S is the sedimentation coefficient, V is the velocity of a particle, and A is an acceleration. So you think about it like you're centrifuging particles, and those if when you're centrifuging the particles, they will settle on a different level. So you have to for some you have to centrifuge at higher speed, some you have to centrifuge at lower speed. So when it settles, that's what the sedimentation coefficient is. So this is in eukaryotes, for example, this is an 80S ribosome, but that means when the ribosome is complete with large and small subunit, the sedimentation coefficient is 80. But when the units are separate, then it's sediments is at 60 and this at 40. So it's not the coefficient is different from the total. It's not the total sum of both. So it's not 60 plus 40 is 100, because when they're together, these units will sediment at 80, not at 100. So just so you know what the S means. Okay, so in addition to those to ribosome small and large subunit and initiation factors, we also need Shine-Dalgarno consensus sequence. The Shine-Dalgarno consensus sequence is upstream of the start codon. The sequence for Shine-Dalgarno is AGG, which is a conserved sequence in prokaryotes. In eukaryotes, there is no conserved sequence like same as Shine-Dalgarno. There are sequences that we will talk about, which are called Kozak sequences, which are not as conserved as the Shine-Dalgarno sequence in prokaryotes. This Shine-Dalgarno sequence is upstream of the start codon. So if this is the start codon, this is upstream of that start codon. So if this is the start codon, here is the Shine-Dalgarno sequence. Shine-Dalgarno sequence is it's a complementary sequence to the sequence on 16S rRNA, ribosomal RNA. This is the smaller subunit RNA. So this small subunit ribosomal RNA will bind to this Shine-Dalgarno sequence in mRNA. So that way mRNA and rRNA will stay together, stay attached, or they will find where the initiation site is. So this is the ribosomal binding site, 16S RNA. This is mRNA. This is start codon. This is 30S subunit. That's where going on. So this is the ribosomal binding site or Shine-Dalgarno sequence. This is in polycistronic RNAs. This is upstream of the ORF. In addition to this ribosomal binding site, the Shine-Dalgarno consensus sequence, we also need a special tRNA. It's called fMAT tRNA fMAT. This fMAT tRNA fMAT is the one that brings the first methionine to the start site. So AUG, that will be a methionine. This methionine, the first methionine, is brought by a tRNA called tRNA fMAT. Instead, and then any other methionine within the sequence will be carried by a regular tRNA. So the first prokaryotic methionine is always N-formal methionine. So this is methionine, this is N-formal methionine or fMAT. This fMAT will be added at the initiation codon, at the initiation site. Okay, this the tRNA fMAT binds normal methionine, which will be within the sequence. All the methionines within the sequence will be carried by, will be brought by this tRNA fMAT, not the fMAT tRNA fMAT. I'm sorry, this is tRNA, it should be tRNA mMAT, not tRNA fMAT here. I'll fix it. Okay, so the very first AUG, this the tRNA that delivers this methionine to internal AUG codon is different. It's tRNA mMAT, not the tRNA fMAT. This fMAT tRNA fMAT is recognized by pre-initiation complex, but not the methionine tRNA mMAT. That will not be recognized by the pre-initiation complex. Okay. Now let's talk about the steps for initiation in prokaryotes. Remember we talked about these three initiation factors. These three initiation factors comes in place first. So this is ribosome 50S and 30S in prokaryotes. It's a 70S ribosome. This ribosomes need to be dissociated first, and this once this dissociation happens, IF3 initiation factor 3 comes and joins at the at this E site of the smaller subunit, and it will keep these two dissociated. It will keep these away because of presence of this cannot bind. Once IF3 is bound, then IF1 will come and bind at the A site of a ribosome, and then IF2 will it's already associated with GTP, and it binds to IF1. So IF1 and IF2 join the 30S IF3 complex. So this is the 30S IF3 complex. These come and join later. IF2 is bound to GTP and is associated to the complex through IF1, which binds to A site. This 30S complex now, whether it's with or without fMAT tRNA mMAT, binds to the mRNA. So most research suggests that fMAT tRNA binds first to this complex, and then it binds to mRNA. Here's the mRNA. Now this here in this figure, it shows that fMAT is already bound here to the IF2 initiation factor 2 on the ribosome, and then this mRNA comes and bind, and remember mRNA comes and bind at Shine-Dalgarno sequence. So a portion of 30S rRNA is complementary to the Shine-Dalgarno sequence that will hydrogen bond there. Position this, and this hydrogen bonding at Shine-Dalgarno sequence will position this fMAT, tRNA fMAT over to the P site. So this is the P site right here. This will be brought here at P site. all the subsequent amino acids will enter at the A site, because there is no other amino acids now to bind to this one. This is the first amino acid, so this brought directly to the P site of the ribosome. This initiation factor 2, it ensures that the tRNA FMAT is the only tRNA that will fit in this complex. So if there is a MMAT tRNA, that will not fit in this will not fit in this position. So that's why the special initiation tRNA is the only one that can fit. Once this sequence is once this pre-initiation complex is formed, then this IF3 will dissociate. This dissociation of IF3 will allow large subunit to bind. So IF3 dissociate, 50S subunit enters, 50S subunit binds here. This GTP has not yet hydrolyzed. It's still bound to this initiation factor 2. This is FMAT. This is initiation factor 1. This is the whole complex now, the initiation complex with the ribosome there. The first amino acid is here. mRNA is bound to the ribosome already. And this is here now. This is the pre-initiation complex. Once this large subunit binds here, then the GTP on IF2 initiation factor 2 will be hydrolyzed. This allows the IF2 and IF1 to dissociate. So these two will dissociate. They have already delivered this first methionine in its correct position here, and this will now be the initiation complex. So next amino acid will come. This is A site is now ready to accept the next amino acid tRNA complex. The next amino acid tRNA complex will come and enter into the A site of the ribosome. Okay. Initiation in eukaryotes is a little more complicated than prokaryotes. So the mechanism is quite similar to prokaryotes, but since mRNA structure is different in eukaryotes compared to prokaryotes, so the initiation is a bit different as well. So similar to prokaryotes, there is a recognition sequence to promote ribosome binding, and initiation factors are necessary to assemble the complex. Some steps are different. The small subunit is recruited to the 5' cap of mRNA. So remember, the eukaryotic mRNA have 5' cap, and the small subunit of ribosome is recruited there at the 5' cap of mRNA. The initiator tRNA, it binds small subunit before the small subunit binds mRNA. In prokaryotes, it's usually the initiator tRNA or the FMAT binds first, and then mRNA binds to small subunit. In eukaryotes, it's opposite. There is no Shine-Dalgarno sequence in eukaryotes. First methionine is not modified, so it's not informal methionine like the eukaryotes, like the prokaryotes. Sorry, so it's not modified. Instead of modification, it is carried on a specialized initiator tRNA. So the specialized initiator tRNA will be the one that will bring this methionine. The components in eukaryotic translation are again ribosome. This ribosome is different. It's ADS, so it's larger, bigger than the prokaryotes. The large subunit is 60S and small subunit is 40S. Initiation factors. Initiation factors in eukaryotes are denoted by EIF, to say eukaryotic initiation factor. There are multiple initiation factors in eukaryotes, so not just the 3 like prokaryotes. So initiation factor 1A, these binds near the ribosomal P site. IF1 binds near the ribosomal P site, while 1A binds near the A site. So remember A, P, and E. The binding to A site is similar to binding of IF1, and binding of initiation factor 1 is similar to binding of initiation factor 3 of prokaryotes. Eukaryotic initiation factor 2 is the main protein complex, which is responsible for delivering the initiator tRNA to the P site of the pre-initiation complex. It requires GTP. So again, somewhat similar to IF2 of prokaryotes. Initiation factor 3 promotes dissociation of intact ribosome and binds 40S subunit, so similar to prokaryotic initiation factor 3 as well. Initiation factor 4 is an adapter. It recognizes the 5' cap. It binds to these PABs, or poly A binding proteins, which is on the 3' end. So it's bind both 5' and 3' end of the mRNA. There are multiple subunits of initiation factor 4 that we will talk about. A, E, and G, for example. Initiation factor 5 promotes 60S subunit binding to the smaller subunit. 5B binds GTP, and it helps initiation factor 2 recruit initiator amino acid tRNA complex. Initiation factor 6 is similar to initiation factor 3. It binds 60S subunit, and it prevents reassociation. In addition to all these initiation factors and ribosomes, there's Kozak consensus sequence, which is similar to prokaryotic Shine-Dalgarno sequence, but it is not as conserved as the Shine-Dalgarno sequence. It's a consensus sequence. MET tRNA initiator MET. So this RNAi doesn't mean RNA interference here. I means initiation. The first eukaryotic methionine in eukaryote is not modified, but is carried on a special tRNA initiator tRNA. The internal methionine are carried on a different tRNA, so tRNA M methionine. So here you can see that the tRNA are different for two methionines. So you can also this will also suggest that eukaryotes have 21 tRNA instead of 20 tRNA, because there are 2 for methionine, 1 for the initiation methionine and 1 for the subsequent methionines within the sequence. This is a really good article because this suggests, in biology there are always exceptions to the rule, and this is an exception where that the initiation can occur in eukaryotes without the initiator tRNA. So if you want to read more about that, this is a really good paper. Okay, so let's talk about the mechanism, the steps, different steps, how initiation or pre-initiation complex occurs in eukaryotes now. So this is the ribosome, large subunit, small subunit, A, P, and E sites. We will talk about this A first, and then we will go over to B. Here, initiation factor, eukaryotic initiation factor 1, 3, 5, 1A, these all come and bind. 1 binds to E site, 1A binds to A site. This IF3 kind of binds to 1 as well, to the site E, and then 5 binds to E site through initiation factor 1. So here 40S ribosomal subunit binds initiation factor 3 at E site. So here this is initiation factor 3, this big molecule here. This binds at the E site. It keeps it dissociated from large subunit. So large subunit dissociates here. 1A binds to A site. E site is bound by these additional initiation factors. Initiation factor 5, 3 is already bound, and 1 as well. So all these bind to this E site. Initiation factor 2 and associated already with the GTP. So this is the IF2 GTP complex. This bind to this methionine, the initiator methionine. tRNA I is the initiator MET. This is methionine here. This bind to this make this complex, and this will assist in this initiation factor 2, because it's bound to this methionine. This will assist the binding of this 40S IF3 with this molecule. So here it binds to IF3. Here the 2 binds to IF3 brings this here puts it in the position at site P. This is the pre-initiation complex in eukaryotes. Now, this methionine is already there. Now the mRNA needs to come and bind here. Okay. Initiation. This is bound here. Now let's look at here. So this is mRNA. mRNA is bound by initiation factor 4. This is the 5' cap. So this is all this binding happen at this 5' cap here. So initiation factor 4, different subunit of initiation factor 4, E subunit, G subunit, and A subunit. They come and bind to these to the 5' end close to the 5' cap. Here, this is the upstream region of the 5' UTR. So that's where all these initiation factors 4 binds to the mRNA. Remember, it also binds to these poly A binding protein or PABs, and this holds mRNA in circle because the PABs are towards the end, towards the 3' end. So it will kind of bring here, and I will show you that in the next set of slides. So here. This is the initiation factor 4 bound to the 5' cap, as well as here in the 5' UTR, and it also binds to these poly A tail proteins or poly A binding proteins, so keeping this mRNA in circular form. And the reason for that is it's because we are not just making one copy of this mRNA, right? We are making multiple copies of mRNA, and that means this, if there's a higher translation or higher expression of that gene, we will be making multiple copies. If this is all circular, once it reaches to the end here, the translation reaches to the end, to the stop here, it dissociates and immediately associates here, so it doesn't take long time to find the mRNA start site again. Okay. So here the initiation factor 1 assist 4DS subunit. So here we have this, we were just talking about all this. It helps 4DS subunit complex to move 5' to 3'. It scans the 5' UTR to find the first AUG. This scanning requires ATP hydrolysis. This ATP hydrolysis is carried out by initiation factor 4. So initiation factor 4 provides that ATP hydrolysis. The Kozak consensus sequence assists in pausing the complex over the first AUG via the hydrogen bond. So this is this IF4 is bound here. This initiation complex is scanning for AUG. It's find the AUG, and that at that site this methionine will bind to that anticodon for AUG. On this methionine will bind to the codon on the mRNA here. Initiation factor 2 hydrolyzes its GTP. So this is IF2 with GTP here. So this will hydrolyze its GTP. This hydrolysis of this GTP will cause dissociation, similar to prokaryotes. So these EF1, initiation factor 1, and initiation factor 3, initiation factor 5, these all will dissociate. Once these dissociate, this will allow the large subunit to come and bind. And before that, initiation factor 5b GTP enters the complex. This that will bridge tRNA and initiation factor 1a. So initiation factor 1a is here, tRNA is here. This will bring those in close proximity. Large subunit then binds. It activates the GTPase activity of this 5b. This 5b then uses that GTP, becomes 5b GDP, and F1a, these dissociate. So, these the 3, 5, and 1 has already been dissociated. Then 1a and this 5b, it's not shown here, that will dissociate this dissociation is because carried out by this GTPase activity of the large subunit. So that means the ribosome working as an enzyme. So ribosome is also called the ribozyme, right? Okay, so now let's talk about this. So we talked about the initiation, pre-initiation and initiation complex. Now let's talk about elongation. So once that first methionine is added on the P site of the ribosomes, either by a specific methionine that has been modified and carried by the same tRNA as the tRNA will carry the methionine in prokaryotes, or the methionine is not modified but it's carried by a special tRNA in eukaryotes. Either way, once that methionine has been added on the P site in ribosome, and the ribosome is now the complete ribosome, large and small subunit. This first methionine is already added. The next step is elongation. And elongation, it's very similar in both. The events are similar in prokaryotes and eukaryotes. There are 3 steps of this elongation process for translation. The first step is tRNA delivery. So in tRNA delivery, so here, this is our the first so this shows there's already been multiple amino acid has been added. It's a growing polypeptide chain. But let's assume this is your first methionine, and the next tRNA will come and bind in this A site. So that's the tRNA delivery site. So in tRNA delivery, tRNA attached to the growing chain, so this tRNA is attached to the growing chain is this tRNA that's in the P site, is already in the P site, right? So either it's your first methionine or it's the last methionine of the growing chain. A complex of EF2, or these are called elongation factor 2. The complex of EF2 and amino acyl tRNA, that means already charged tRNA that has the correct amino acid added to it, and GTP. So here, this is amino acyl tRNA, this is EF2 GTP, and this is GTP. These deliver this to this A site. So you can see this EF2 and GTP act as the initiation factor 2, right? Because that has GTP as well. So this elongation factor along with this GTP binds to amino acyl tRNA, brings it to the A site. There is no hydrolysis of GTP yet. This will happen afterwards. Okay, this binding of this entire complex require correct base pairing between the amino acid tRNA and the mRNA in the A site. So the mRNA here, so this that means the codon and anticodon need to be correct. So it's need to be correct base pairing. So whatever the tRNA is coming here will read this codon on the mRNA, and this the proper anticodon will make the complementary sequence here. Once this pairing is established, then this GTP will be hydrolyzed. This hydrolysis happens by a ribosomal dependent GTPase activity. Again, because ribosome is also a ribozyme, it has this activity. And what that does is, once this GTP is hydrolyzed, that will it changes the structure of EF2, or the 3' the tertiary structure of EF2. It changes, it rotates the tRNA amino acid into the correct orientation. So if you look now, the amino acid is on this side. Once this GTP is hydrolyzed, this energy will change this tertiary structure of this EF2. With this EF2 will then rotate this whole mRNA, this portion of mRNA, to bring this amino acid towards the growing polypeptide chain. So here, this is EF2 GDP. This is hydrolyzed. This phosphate is released. This is switched or rotate this position of amino acid. Amino acid is now close to this growing chain of polypeptide. Now this brings these amino acid in close proximity for peptide bond formation to occur. Okay. This is called accommodation. So this rotating of tRNA amino acid into the correct orientation for activity is called accommodation. At this point, EF2-GDP complex will dissociate from the ribosome because this amino acid has been brought in the close proximity. Dissociation of EF2 is necessary before the second step because this EF2 masks the amino acid. See here, this amino acid is covered, is masked by this EF2. So if this until this EF2 dissociate, this amino acid is not available for binding, and so this need to be open, need to be available. Second step is peptide bond formation. So this accommodation results in, so this is just an example here. There is, I think it's a methionine. This is threonine. This comes in. This was originally the EF2 was associated. So this is threonine here. Amino acid tRNA binding to A site. So threonine will be here. EF2 will be bound to this. After accommodation, it switches the size. Now the threonine is here, close to the previous amino acid. The peptide bond formation happens between these two amino acid, the last amino acid of the growing chain and the incoming amino acid here. This peptide bond formation, as this accommodation results in both the 3' CCA portion of the amino acid tRNA in the A site and P site to hydrogen bond with the ribosomal RNA. So it's here. So these are bound to the ribosomal RNA. This orients the amino group of amino acid tRNA to the carbonyl of polypeptidyl tRNA. So this is the carbonyl group of the previous ones. This is the amino group of the incoming one. So these can make the bond. Peptidyl transferase is an enzyme that will catalyze this formation of peptide bond between the peptide in P site and the peptide in A site. The one that's newly arrived amino acid tRNA in the A site. This will lengthen the polypeptide chain by 1. Right, so it's 1 every time. This peptidyl transferase is catalyzed by the RNA in large subunit. So again, as I said, ribosome is a ribozyme because it has catalytic activity. And so here again, this is the ACC site. That's where the amino acid is bound. Okay, so now once these two amino acids, these bind, so now this is the one that's already bound. This is the new one that's in the P site, and then the next amino acid that comes in A site will bind to this amino acid now in threonine. The one that was in P site before, that will go in the exit site, it will be pushed to the exit site. So this is the second step, is the peptide bond formation. Third step is called translocation. Translocation, an elongation factor EFG. It's instead of EF2, this is EFG elongation factor. This EFG is bound to GTP. So this is EFG bound to GTP. This recognizes a change in the conformation of the tRNA in A site, which is bound to the peptide. And it binds there. So these are the two. This is the one that was already in the P site. This is the incoming amino acid. These two made this peptide bond formation in the previous step. This EFG will recognize a change in the conformation of the tRNA in the A site. It's right here. And this will bind to this, the one in the A site. So this will bind to this one. This binding will stimulate GTP hydrolysis because the EFG contains this GTP. This GTP hydrolysis will then release this energy. GTP converts into GDP. And then it will change the EFG conformation. It will open the gate between A and P site. So this will be then pushed into the P site, or it will be forced into the P site. Now the peptidyl tRNA with its mRNA codon in the A site is now in the P site. So this one will be pushed into exit. It will come out and exit. So now this is the next one that will bind to the one coming in A. So another very good paper here about process of this mRNA tRNA translocation, if you want to read more about translocation. And this is the last slide for today, except for this video. This is one of my favorite scientific video. It's really old, older than me, much older. So it was this was made in 1971 at Stanford University. It's a really neat video if you want to watch. This goes into all the initiation factor, how those initiation factors binds and start the initiation of protein synthesis or translation. So it goes through all those hydrolysis of GTP and ATP, and it's really nice. Okay, and with that, this is the end of this lecture. We will continue talking about translation in the next lecture, and there will be a quiz today. All right.